Welcome to Data in Biotech, a podcast from Cordyne where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. In this episode, we sit down with Ilya Goldberg and Reese Findley from ViQi, a company at the forefront of high-end imaging and AI solutions for biotech. They shared the story behind ViQi's beginnings, dive deep into their groundbreaking imaging technologies, and how machine learning is revolutionizing the analysis of cellular images. We cover advancements in live cell imaging, lab automation, and how AI is being trained for viral infectivity assays and drug discovery. Ilya and Reese also talk about the future of AI in life sciences and its potential to transform lab workflows and accelerate scientific breakthroughs. Here we go.
Ilya Goldberg and Reese Findley, welcome to the Data in Biotech podcast.
Hi.
So just to kick us off, starting with you, Ilya, and then to you, Reese, would you just give us a brief introduction to your background and what brought you here today?
So my background is computational and life science. I have a PhD in life science. I ran a research lab that was hybrid at the NIH for almost 20 years. I joined ViQi about 6 years ago. Before ViQi, I did a startup as a medical device that used AIs to predict cancer risk and lung nodules from CT scans.
Awesome. Reese?
My background is in neuroscience. I did my PhD in olfactory research in mice and studying the neural correlates. And I now work for ViQi as an AI data scientist. And I do drug discovery and phenotypic profiling using automated AI tools.
Awesome. Since you're both at ViQi and we're here to talk about ViQi today, would you all give an introduction to ViQi and the kind of work that you do?
Yeah, so ViQi started actually in the late 90s as an open source project that you see Santa Barbara, the computer science department, as a collaboration with several biology departments to basically address high-end imaging needs. So multi-dimensional microscopy, like 3D, multi-channel, large sets of images. So it was a project to build infrastructure to support this, and obviously analysis.
It sounds like a lot of what ViQi does is related to imaging and using imaging and machine learning for different types of problems that you're trying to solve. Can you just give us an overview of the way that imaging and machine learning or AI are applied?
Yeah, so I would say that imaging is really central to the company since its beginning. A lot of us started using AIs to solve imaging problems, I would say in the early 2000s. So we started building our first AIs to do this kind of thing. It was readily apparent really early on, even with very primitive AIs, we're using like perceptrons, which are like not deep neural nets, and other conventional sort of classifiers. And even with that, we can outstrip experts in imaging. So people who interpret images professionally every day, even that primitive of an AI could surpass what they could do very routinely. So that, of course, told us, holy cow, you can do a lot with this. So some of us started way back when really exploring the space and moving into like not using stains, so using brightfield microscopy. So you're using a much wider sort of net to measure things about images. It was very clear to the people who started playing with this technology early on how like disruptive and pivotal it was going to be in the future because the tools were just so immensely powerful so quickly right away.
You've been dealing in both imaging and AI for a long time now. I'm wondering, where do you see the combination of imaging and AI as having made the greatest contributions to biology and to the types of work that you do?
In the broader scheme of biology, technological development has been booming in several areas. We're seeing live cell dyes so you can capture time courses. We're seeing better resolution automated imagers. We're seeing faster automated imagers. And then we're seeing these AI developments. And I think the biggest question that's being answered right now is what can be automated and what can't. And so I think that's what ViQi does really well, actually, is what is appropriate to automate and what is very difficult and really needs that subjective expert science opinion behind it. And I think that's where the most advances have happened in the field in general, is taking those areas where you can automate and just shortening that pipeline down, reducing massive bottlenecks that have been done manually for decades down to hours that an automated imager can do with an automated analysis system.
Yeah, that's a really interesting point, and I would love to hear from you. What have you learned about the boundaries between what can be automated and what can't be automated in terms of the type of work that you do?
I think in my work specifically, I work on our assay, automated high content screening. And one thing that we do is we do a type of phenotypic profiling where we train an AI on a bunch of different compounds that have been applied to cells. And we use the distances to create this distance-based dendrogram. And all of that works really well. I think that we usually get really cool biological meaning out of it. However, it demands a biologist to look at the final product. And the reason I say that is because this clustering is subtle and it's stochastic. And anybody who's worked in clustering, I used to cluster neurons. Anybody who's done that knows that different people can get very different results depending on the analysis methods that they're using. And it's a huge problem, especially in sensory neuroscience. And that requires that sort of expert eye, some bioinformatics validation of our clusters. There's a lot of now advanced science that goes into interpreting the end result of our automated analysis.
Yeah, it does. And I'd love to dive into AutoHCS in just a moment. But before I do, can you just help orient me to how ViQi partners with clients or customers, and what's the type of work that you do in support of the biotech ecosystem?
We actually solve two different problems. So one of them actually is related to your last question about automation. One of the big things that we have to deal with that's not automated is the interface. So, you have lab, you have an imager in the lab, and then you have all these high-end tools that are online on the cloud to analyze this. So every time you move this stuff around, so your experiment down to an imager's images up to the cloud, there's an interpretation. What does all this mean? So adding metadata to your images, specifically dealing with image formats. Everybody's got their own image format. In life science, there's over 100 of them. Dealing with that's actually a lot of where automation breaks. For instance, describing your experiment. The imager knows how it took the image. So you can query the imager, like what's the exposure time, etc. But what compound is in each well of a plate? That's up to reading it out of a spreadsheet or sometimes out of a screenshot of a spreadsheet. So all of the various ways of breaking that have been done. And that's where actually we spend a lot of time.
I should think of this as assay as a service, that yes. A life science practitioner or a biotech organization recognizes the need for an assay and they believe it. I guess
we call it that. It has an unfortunate acronym, but we do call it that.
Yes, it does. But I think the question that comes to mind for me is, how does someone know that the type of assays that ViQi provides are the assays that they're looking for versus the other assays that they might develop in-house, or the assays that they might be able to contract with other people to create?
Anybody who's worked in labs for a long time will tell you there's so many steps that are just aggravating and frustrating and make you want to pound your head against the wall. And some of us pride ourselves in doing those. That's a scientist-like thing. Mine was calibrating airborne olfactory stimuli. Super hard. But anyway, there's something that's just absolutely aggravating that takes a super long time. And to be honest, I don't think it crosses most people's minds to try to automate those steps. What they want to automate is their image analysis afterwards. And that's what they're talking to their automation teams about. What ViQi is doing all the time is trying to identify those just hair-pulling steps that are taking too long and causing too many issues, and find a creative solution to automate that out so that people can do the fun part of science. And so I think when you ask what are people coming to ViQi for, it's those creative solutions, because we're spending all of our time thinking about those as opposed to trying to do it while we're trying to execute bench experiments and meet deadlines, et cetera.
That makes sense. Before we dive into an automated viral infectivity assay, could you just talk about what are the uses for a viral infectivity assay broadly?
So it's surprisingly broad use. But anytime you have a research lab or manufacturing or vaccine sort of development, you need to measure how much virus do I have? How much infectious functional virus do I have? In vaccines, for instance, you're looking at a population that's been immunized with a vaccine and new variants come up. And you want to know this population, are they resistant to these new variants? So you need to grow up your new variant, hopefully before it becomes pandemic level. And you need to collect blood samples from your patients and measure the inactivating antibodies that they have in their serum that basically inactivate the virus. You're measuring how much virus do I have left after I treat my starting virus with a vaccinated person's serum. That's like vaccines. Of course, to get to vaccines, you have to manufacture a lot of virus. And during the manufacturing process, you're growing up cells. Normally, viruses are grown in cells. So you need to measure how much did I get, because of course, it's all biology. So it's different every day. Things fail. You need to measure that as early as possible because it's a weeks-long process to grow this stuff up in vats and purify it so that you can inject it into people. Huge number of steps all along the way from R&D to normal manufacturing process. You're always running these assays. And of course, in R&D, you're studying viruses for their et cetera. You need to know how much virus there is. So it's a absolutely ubiquitous assay in virology.
Yeah, that makes sense. So what are the traditional methods for conducting this type of assay?
So normally those methods rely on enough cell death that you can detect it easily. Okay, so viruses normally kill cells. If you have a cultured cells that are living on a lawn, on a plastic dish, what you can do, so this is a plaque assay. Basically, you dilute your virus enough so that you just have a scattering of individual viral particles that are infectious land on individual cells. Then you wait long enough for the neighboring cells to become infected so that you get a little ring of cell death, a hole in your continuous lawn of cells. That's a plaque. And then you sit there and count them. And that's what I mean is people are like, why am I sitting here counting plaques? Shouldn't I take a picture of this and get some software to count them for me? You're solving the very last step of a problem that you could have solved a lot earlier. Why are you waiting all this time for multiple rounds of infection? Why rely on cell death for your output?
Interesting. So in the traditional plaque-based assay, the images come at the end.
Yeah.
And then there's a counting that occurs.
If at all.
Whereas with AVIA, if I understand correctly, one of the things that you're doing is you're taking images at the beginning, the middle, and the end. And
well, we're taking them at the beginning. So we're saying instead of detecting cells when they're good and dead, we're going to detect cells that are producing virus much earlier in the cycle, where cells have to be healthy in order to produce virus. Otherwise, they're not going to do it, right? So they're very much alive and healthy, and they're cranking this virus out. And viruses have evolved to basically hijack cellular process to make copies of themselves instead of the normal business of the cell. So that changes the way the cell looks. Those changes are very subtle. There's a lot of variation in how cells look like. So it's a perfect problem for an AI to solve because there's a huge amount of variation. It's a noise filter is what you're relying on it to do. There's a lot of information. A lot of it's irrelevant. You need to focus the AI in. This is what infected cells look like. This is what uninfected cells look like. Now go and comb through these images and count areas that are infected versus not. And that's what the assay is based on. And the other thing that people normally do with microscopy of cells is they use antibodies or other kinds of molecular probes that basically highlight very specific molecules in these cells with fluorescent dyes so that you can just detect infected cells because you're detecting viral proteins that normally don't exist in cells unless they're infected. That's another kind of assay, but you don't have to do that with Avia because there's no probes, there's no specific detection of specific proteins. You're broadly just asking cells that are infected look different from cells that aren't. And this is something an AI can tell very easily. And you can look at these images, and if you can see anything, you certainly are not going to be able to quantify this and turn it into account.
Going back to what Reese was mentioning earlier, it strikes me that the methodology that you're approaching, yes, you have the images that you're taking that can, and then you have the AI that's consuming the images that can detect very early these very subtle changes in the cells that indicate that virus is going to be produced. But then there's this automation and design of experiments component that allows you to feed sufficient information to the AI model that it's able to detect and understand the level of virus that's happening inside of the cell based on it. So I would love for you to just explain sort of the methodology of how that comes together.
So by design, this is tied into high content screening. So it's a good segue into that also, because it all relies on equipment automation. So people have developed automated imagers really since the mid 90s or so. We were there early enough where we went to Olympus and Nikon and all these microscope manufacturers and say, hey, you know what you should do is you should have a robot essentially move this plate around, do autofocus and image cells. And people are like, the hell are you talking about? People use cover slips and they sit there and meticulously manually image these things. And, the first microscopes for high content screening are made by hand to basically put robotics X, Y, Z positioners on these plates. These plates are like standard sort of form factor. They're like this big, 3x5
96
inches.
wells, typically,
Yeah,
384 wells. Yes,
exactly.
yeah.
Yes. That's the same format, except now you're growing cells in the bottom of each well and you're putting drugs on them or a genetic manipulation library or whatever. And so that's the base of the experiment. Now you need to acquire millions of images, potentially a stack of plates that you want a robot arm feeding into your microscope and taking them out of the incubator and putting them back. So you need autofocus is a key component of this, somewhat precise stage positioning to get to the right places in all these wells. So that's the automation. And then upstream from that, of course, if you have a chemical library with 1 million compounds, you have robots already that deal with pulling compounds out of the freezer, arranging them on plates, collating cells, doing all of this kind of stuff. That's already existing. So part of the development of Avia was, in fact, very specifically to plug it into the HCS infrastructure. So you don't have steps that are not easily done by robots. Like a plaque assay requires an overlay, so you have to pour like a gel over this thing. That's really hard to handle because it has a narrow temperature range, blah, blah, blah. Anytime you add a step, you need to stain the cells or wash them or something when you're dealing with dozens or hundreds of plates. It's a huge multiplier for how much time, effort, reagents you need for this. So all of that was a process of what can we get rid of to increase automation? Because automation is really the key to this, right?
Right, because of the speed and efficiency with which you can get the results, right? So you can get the results in a much shorter period of time and at much lower expense than you could
Not
otherwise.
only that, so those are really important properties of automation. A thing that a lot of people skip is precision and reproducibility,
yes,
which you get from automation as opposed to people manually pipetting and adding reagents and stuff. So pipetting mostly. If you can get robots to do the pipetting, you get better precision. We detect, because our tools are so precise, so we can routinely detect differences in people's pipetting techniques and recognize that this person is typically low readout on the assay, whereas this one is consistently high, or they have different levels of variation because people are different. And robots, you can eliminate a lot of those differences. So
my understanding is that you have this automation that's taking images of each of these wells, and what you're putting in each of the wells is designed to provide the controls and provide the degree of infectivity that sort of characterizes the distribution that the model needs to pick up on.
It's important to think about 2 stages. The first stage is really training. You have to train the AI. You don't need to train it in every experiment, but if you train an AI that can be used across experiments, in life science, every time you run an experiment, things are slightly different. So you have to train your AI across these differences, not just cell-to-cell differences, but experiment-to-experiment differences. But you can do this, and that's a reproducible AI. Now that you have it, now you can feed it an experiment. There's very different modes of operation for training and for processing. So it's important to recognize that because you're doing things very differently. In training, you need a lot of repetitions. You need a lot more data. You need the AI to be exposed to a lot of examples. When you're processing, you need a lot less data because the AI will give you an answer for each individual little piece of an image or each individual cell.
Makes sense. My understanding is that one of the benefits of a VIA is that you can apply it to any number of viruses that might require a viral infectivity assay, and so it's broadly applicable. And when you talk about the difference between training and inference, what's coming to mind for me is, are you having to go through a separate training process for sort of each type of virus that you're running through the assay?
Exactly. So that actually has to do with automation of AI training, because instead of trying to come up with a universal AI that recognizes infected cells, instead we're like, different viruses and different cells have very different interactions. And so they produce very different phenotypes. And so we dropped that whole idea and said, no, we're just going to train an AI for each lab. So even if we work with this kind of virus and this kind of cell line, we're going to train in your lab because there are enough differences lab to lab. Certainly different people use different imagers. The optics are different enough from even the same model imager that you can detect. If you're like, can I train an AI to detect the same model imager in the same lab, just 2 different models? Of course you can. An AI can be trained quite easily to tell you which imager did the image come from. So those differences are definitely baked into the data. And so if you're trying to universalize your AI, you have to train it across the variation you're going to see. Otherwise, it'll focus on the wrong things. And that's one of the sort of caveats of dealing with AIs, is that you can very easily get yourself into a situation where the AI is trained on absolutely the wrong thing. We typically refer to that broadly as bias, and that actually fits with how sort of the general public perceives bias in AI. It's the same. The source of the problem is the same.
My understanding is that the primary modeling methodology that you're using for the assay that you're talking about here are convolutional neural networks, or CNNs. Is that correct? What is the application of CNNs to the way that you do this work?
So we're AI agnostic. So we use different CNNs, but we also use more traditional AI techniques that were called machine learning and classifiers before they were neuronal based. So there's a lot. So we use it's split for not just because of how things started. So AutoHCS tends to be based on features and classifiers. Image features, we'll get into what image features are, but it's more how people approached this in the 2000s before CNNs became very practical. AVIA, on the other hand, uses CNNs, and it uses modern pre-trained CNNs trained on other imaging problems unrelated to cells or viruses. Typically, they're trained on, we are all training these AIs by clicking on images of stoplights and bicycles and cats and dogs.
Are there any particular model families of CNNs that you've found transfer learning is really good for biological images?
We tried a a fairly broad array of these. We tend to use EfficientNet in various versions, trained initially on ImageNet data, and then transfer learning to, typically do direct transfer learning to infectivity problem from weights loaded from image discrimination kind of problem.
I think it's also important to note we're AI agnostic and we're pretty willing to try anything, but we do optimize the AI we use based on the experiment that we're running. And we'll get into it, but with Avia, we tend to want binary answers about little tiny tiles. And so that works really well with CNNs. Whereas with AutoHCS, we're actually using the AI's confusion to give us information about the biological status of an image. And so then that's why we're using feature classifiers, because they're more willing to be confused. Whereas you could say CNNs are snappy. They have very, very specific answers that they want to give, and either confidently correct or confidently wrong. Like we're always doing development. We're always testing how different AIs respond to our experiments. But we find that there is optimization to be had depending on what type of question you're asking.
I'm interested in how you think through the selection of the players in the ensemble when developing an assay like that.
That's an important feature of AIs that people don't appreciate enough, I think, is that they are stochastic. So if you train an AI with the same data set and then ask it the same question, you're going to get different answers, keeping everything exactly fixed. And that is because during AI training, there are processes that go on they're randomized. Like in a GPU, for instance, all of these things are happening at the same time. That sets up like a zillions of race conditions. How the race is resolved is essentially random. You can do this in a completely deterministic way, but you're essentially serializing the process. It makes AI training impossible. So every time you train an AI with the same data, you're going to get a different AI. So that's one reason why you would have essentially a voting kind of model for an answer that is subtle enough. That's because a lot of times AIs just get the right answer, and why would you get another take on it? It's pointless. But there are enough problems where the answer is not well determined, or the AIs are just not able to do this well enough. But if you have a group of AIs that vote, even if it's the same AI, that you'll get a better answer, because it's just statistics, okay, help you get a better answer. But the other reason to use it is because different AI models, they're going to be biased in different ways, and they're going to give you different answers. And we typically actually don't average the results from these ensemble. When we do this, we always see that the AI result is always better than the average.
Yeah, that makes sense. So you have an ensemble of models. Should I think of that as multiple CNNs trained with different hyperparameters, or the same
models,
different model types? It sounds like you also bring some traditional computer vision type approaches to bear. Are you incorporating those into the ensemble? And then obviously on top of that, you've got some sort of potentially like a decision tree model or something like that sits on top of it that sort of uses the information from the different models to make a decision. Am I thinking about that?
Yeah. So with CNN, so typically the input to a CNN is pixels. So the input layer of neurons is really pixels, raw pixels, or normalized pixels. And then you train the AI model from that. But you're not limited to that, because you can put tabular data into the input layers also, in addition to the pixels, because it doesn't know what's a pixel, what's a cell in a table. So you can combine the results from conventional image analysis, which typically comes out as tabular data, together with your pixel data. We do this all, I think we have done this more as conceptually easier with feature-based machine learning and classifiers, because the input is a table. So you're just going to say some of this table comes from algorithms that process pixel data and turn it into table numbers, and other stuff is genetics or pharmacokinetics or whatever. Physiology, what's your blood pressure? Because that all becomes tables of numbers, and then it just churns through so it's similar idea. We call this multimodal analysis because part of your data is image based and part of it is not.
Yeah, that makes sense. And this is how a lot of the biological foundation models are trained right now too, is with different sort of multimodal data sets concatenated together and then brought together in order to get the signals that you're looking for. Reese, can you just give us an introduction to high content screens and how they're used broadly? And then what is AutoHCS and what do you bring to the table in that?
Our automated high content screening tool is used to analyze high content screens, which are screens usually, not always, but usually collected on automated imagers. And they are high content because they generally have either brightfield images or several channels, and you can pull a lot of information out of them. And specifically, if you have AIs, you can pull a lot of information out of them. And so our automated high content screening tool it actually has expanded quite a bit, but it started out to analyze these drug discovery assays where you apply a lot of different compounds at a lot of different doses to a series of cells and you see what they do. And we train a series of AIs. This kind of layers on top of AVIA, uses controls versus infected cells to train an AI to determine the difference between two. Right. And we use controls with AutoHCS too, except in this case, we are looking at target phenotypes. And so we have a negative control versus, let's say, you're interested in apoptosis. You have a certain dose of starsporin as your positive control. And now you have a identified target phenotype. You know what it does, when it does it, and you can compare other test drugs and see if they do the same thing. You can also train AIs on dose and get dose response curves. And those are extremely valuable because some compounds have a very binary response. They're either on or off. Some, actually most, have induce multiple phenotypes depending on what dose they're at. Usually at the highest dose, it's some sort of extreme stress or death. And so we expect to see several clusters with those types of compounds when we train on dose. And then finally, we train what we call the all compound AI, which is what I was discussing earlier, which is an AI trained on every compound at a specific dose. We can do either the only dose that's on the plate, we can do a highest dose. We also have a mechanism of training AIs to determine the lowest effective dose according to the AI. So basically that's the first time the AI thinks it looks different than the negative control. That's the lowest dose that anything happens according to the AI. And so that's what we determine as I changed the name to lowest detectable dose since effective has some connotation to it. So that's the basic framework of AutoHCS. And that's what we've used to analyze screens that are manually collected with 3 compounds up to 1,500 compounds. However, we have expanded this toolkit to also do experiments that are a little more complicated. For example, we've done background screening where you're, for example, looking at neuroprotection. And in this case, there's a compound applied that induces peripheral neuropathy, let's say, on neurons. And then there are protective compounds applied, or at least test protective compounds. And you want to see how much does this now look like the negative control where I didn't apply anything. And we've had pretty good success with identifying test compounds that are protective in that case. We've also looked at stem cell differentiation, where stem cells are put into 2 different growth medias, and then where do they differentiate? What are the stages of differentiation? And our AIs have been able to determine differentiation across a time course at the key stages that we would expect. I guess that leads me to one of the great benefits of AutoHCS is we have the capacity to analyze across time courses. Something that we talk a lot about is analyzing in brightfield. The brightfield images actually contain quite a bit of information, and I think people are realizing that now. People are starting to analyze in brightfield, but the benefit is that you can now take a time course. You're not killing your cells. There's also live cell dyes. One of our big collaborators is Saguaro Technologies. They develop live cell dyes, and we do analysis of their test conditions over time courses. And because we have such an optimized system, we're able to train AIs very quickly. So I've been able to train AIs at each time point across a massive screen and have it take a couple hours. And so that's I think becoming the key benefit of AutoHCS is our capacity to just rapidly train and accurately train AIs and then be able to analyze the data. So there's a lot to talk about in the toolkit, and we can focus on any individual aspect, but that's a broad overview of everything we've done with it so far.
Yeah, I love it. And you're right. There's so many places to touch on. Where I'd like to go from here is just understanding from the time that you receive imagery and you have a problem defined, how do you think through designing the way that you collect your experimental data or the way that you structure sort of the modeling and the assay process to get the signal that you're looking for?
Primarily with AutoHCS, I tend to use feature classifiers. We have our own version of image feature extraction where we're extracting about 2,000 to 3,000 features per image, actually per image tile, because we tile them down. And when considering that, with feature classifiers, we don't need quite as much information or data as a CNN, especially since we are, again, using that AI confusion to give us information about how close or how far apart in phenotype 2 compounds induce. And we can do this analysis on smaller plates. The benefit is that we can increase it to a very large size, but we actually can do this analysis on, I think our absolute minimum is recommended 12 images per condition. Although we have done less just to say, let's just see what happens. But we recommend about 12 images per condition, and we design plates with the scientists usually. We can receive plates that already have data on them and we figure it out. But it's nice to be able to sit and design the experiment with the scientists because you want to consider, are you interested in dose response? Then you do need to save a fair amount of the plate for dose response so that we can get enough replicates of each dose in order to give you good information about it. Are you not interested in dose response? Okay, let's just leave that to the side. Let's maybe put 2 doses on each plate and let's focus on having replicates of each of your compounds. Do you have an unlimited budget? Do it all and we'll pick what we want to analyze. And so that's the initial conversation that I'll have with collaborators is, what is your experimental question? What are you interested in answering? And I think a big benefit of ViQi is my background is not in computer science formally. I'm a biologist. So I can sit and have a very high level conversation with our science teams and then go and do the technical work.
I loved where you were going in terms of how the dose response question goes. Do your clients generally know in advance what are the phenotypes that they're looking to detect, or are you helping them to determine what are the different positive and negative controls that you want to introduce?
Yeah, it really depends on the client. So some people want to detect a certain type of cell death or cell stress, and they know exactly what compound induces that, and that's going to be their positive control. That happens pretty frequently. They're always going to know more about the compound space than we do because they're in the lab, they've been studying this, they know exactly the phenotype they're interested in. There's also the larger and more general phenotypic profiling where basically you're given a very large screen and they want to know which compounds look similar. I don't actually have a target phenotype. I'm just interested in how all these compounds compare to each other. And so that's where we get into that space where you need to make a decision of what to automate. And so our automated analysis will cluster all those compounds into interesting clusters. But then we actually, she's not here today, but we have a bioinformaticist who will go and do pharmacological validation and go look at open source data sets, look at the pharmacological background of each of these compounds, what's known, and start doing ground truth validation of each of our clusters and making sure that they make sense and to what degree do they not make sense? Because those could be compounds that are inducing off-target effects, which is something people are very interested in. And so if there's a compound that really doesn't fit in the cluster that it's in, that's great. That's exciting. That's the one that we point to and we say, you're interested in this. You should go do some other work to look into this compound. And it really depends on the client or collaborator's needs. And generally, they know best what they need. And I'm able to say, okay, if you are super interested in apoptosis, let's identify 3 positive controls for it. And let's make sure that we're doing our due diligence there. Let's also find a positive control for a different type of cell stress or cell death so that we're making sure we delineate between the two. Those are the kind of questions that I ask to make sure that we're doing our due diligence. But I would say generally, with a little bit of guidance on how the AI works, biologists know what they want and they have a good understanding of what they need on the plate.
Probably the most challenging kind of question is this compound doing anything weird? Because that's most failures in drug development have to do with toxicity. And so the compounds are doing something inappropriate. So that's the off-target effects. So you might have a positive phenotype kind of outcome that I want compounds that do this kind of thing. But you want to compare them to other compounds that do other kinds of things and see if at certain doses or under certain conditions or in certain cell lines, your compound of interest is actually falling into a different cluster. In the worst case, in a cluster of compounds that are known to cause problems. So this
Totally.
phenotypic mapping is the area that is really the new area that is offered by AI, because you're quantitatively comparing similarities of phenotype. That's a very hand-wavy thing. But
I think a great
AIs can,
example.
yeah, they can
Sorry,
quantify
Ilya.
that.
It strikes me that because of the image-based data sets that you're using, there's just so much information content inside of the images that can answer both the questions that you're trying to answer with the assay, but also leaves information on the table for the other follow-up questions that you might want to ask, such as, Ilya, what you're talking about with toxicity and with the ability to cluster things together. You can then take a different angle on the question and compare the images to different things that you weren't expecting to have to compare them to, but because you're starting from that place of working with microscopy, you have the sort of information content available to do that. Am I thinking about that right?
Yeah, and I think along those lines, something that I've thought a lot about in this space is the phenotypic space that the AI is exposed to. So if you have two compounds that do really similar things and you train a binary AI to distinguish these two compounds, if it's successful, which it isn't always, it might be confused, but if it's successful, now in that phenotypic space, those two are going to look really far apart. But now you add a third compound that does something very different, and all of a sudden they get very close together. You're giving the AI a relative amount of phenotypic information. And so what I'm actually literally running it right now, I have a series of time points and a series of compounds, and I started out by combining all the time points and compounds and running a really big AI on it. And then I look at what I think is an interesting cluster, and now I train an AI on just that subcluster so that I can get more space between those compounds and have a better understanding of how they separate in that smaller phenotypic space. So there's exactly like you're saying, there's so much information in the image content, and depending on how much information you give an AI about that, it's going to make determinations based on that. And that really comes into having a good understanding of how AIs train, and what they're exposed to is going to determine how they structure the clusters that you get back.
Are there any case studies that you can share from your client work, the projects that you felt really proud of the outcome that you delivered?
Personally, I really like our time courses. I think they're novel. I think they're interesting. I think that it demonstrates a drug's efficacy over time in a way that these single time points really don't. And that was maybe it was naive of me coming from a neuroscience behavior background, but that's something that I found very striking and it really was a very fast result for me to look at it and say, oh, wow, we should be doing this the whole time. We should be doing time courses with every experiment because the kinetics and provide a lot of information. And so I would say being able to develop that and having it run so quickly, so consistently, I'm proud of it.
I think that specific example of time courses has traditionally been prohibitive, because if you're doing your imaging on fixed cells, which you need to do if you're doing fluorescent probes and stuff like that, the cells are dead. There is no second time point. If you want to get another time point, you have to start the whole experiment again. If you're doing this in brightfield on live cells that haven't been stained, you get the other time points essentially for free, because it's literally a robot that's doing the imaging at the other time point. It's a huge area that opens up. Studying things in a time course is just the only reason really it wasn't done. People know this, obviously. The only reason it hasn't been done is because it's just prohibitive, expensive. So being able to process things in brightfield and these kind of broad, non-toxic dyes is what opens this whole thing up. So I think that's a really good point, because it opens up a whole territory that's really been unavailable by other means.
Yeah. This has been really fascinating. And just as we head toward the end, I just have a couple of wrap-up questions. What excites you most about the future of microscopic imaging and AI in life sciences?
I think for me, it's actually the fact that it's becoming more common, because being at the technology development forefront, a lot of times you wait a long time for the technology to actually become widely used. So to me, I think the future is the breadth of applications. And so there's more brains and eyes involved. Everybody is now thinking, how can I use AI? So the breadth of applications now, I think, is really exciting. And it's really taking off on this logarithmic expansion, and there's a couple of decades where people were ticking things over and you're like, oh my God, this is going to be really great. And then a lot of times in technology development, you're like, okay, when is it going to be really great? So I think that time is now. I think that's, to me, the most exciting part of it is how broadly used it's going to be very soon. And now even.
Reese?
And I think for me, I bring it full circle. I am excited about alleviating frustrations for bench scientists. I think that is such a cool way to contribute to science, because so much of their energy and attention goes towards these frustrating tasks, wherein it could be going towards analysis and innovation. And so I like that that's what our tools do. I look forward to continuing to do that with different assays. And it's really exciting for me when I can come to somebody and say, oh yeah, that thing that took 4 weeks for you and made you want to pull your hair out, let me do that for you. And I can do it in a fraction of the amount of time.
So that actually applies to medicine as well, where you have imaging and medicine, like radiologists and pathologists would be like, aren't I out of a job because my job is basically staring at images and saying what's going on there? But much truer and better answer to that is that radiologists, pathologists now get to do far more interesting work, because most of what they do is like the trained monkey work, and AIs are great at this. They're not great at the sort of corner cases and edge cases of medicine. These are the interesting cases. This is where now a lot of knowledge of medicine can be brought in and become relevant, as opposed to screening through thousands and thousands of samples where the answer is very clear cut. So if you drop that out of their workflow, it actually makes their jobs a lot more interesting than they are now, because they are doing stuff that AI should be doing.
That makes a lot of sense. As we wrap up, for listeners who are intrigued by ViQi or just microscopic imaging-based AI in general, where would you suggest they go to learn more?
Our website is a good place to start.
That's ViQi?
ViQi AI.com.
Yeah. Okay.
Yeah, our website has a lot of our previous publications, and then it also, if you go to our LinkedIn, you'll see announcements of the conferences that we'll be at, and there's contact information on both.
Awesome. Ilya and Reese, it's been a pleasure talking with you today. Thanks so much for joining, and look forward to connecting down the line.
Great. Thank you.
Thank you.
This was fun.
Yeah, nice to meet you.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.